You're listening to The Solution, a podcast by Growers Mineral. I'm your producer, Russell Bobel. With me, as always, is our host, Marguerite Fall. A few weeks ago, we sat down with Dr. Judy Fitzpatrick.
She is a microbiologist and a recognized leader in the development of on-site diagnostic tests. She is also the founder of a company called MicroBIOMETER. MicroBIOMETER is a 20-minute on-site soil test to determine microbial biomass and fungal to bacterial ratio.
Our director of research, Jim Hallbeisen, has been interested in their product for some time now. He and Judy really seemed to click once they got talking, didn't they?
Yes, they really did.
I think if you didn't actually jump in there, they would have spent the whole day talking about soil biology.
They certainly would, and I think we could spend the whole day talking about them, talking about it, but without further ado, let's hear what they had to say.
Hi, Margaret. Hi, Russell.
Hello.
How are you? Can you tell me a little bit more about your business before we start?
I think you having some background is probably a good idea to start.
Okay.
I'm going to hand that over to Jim.
Very good.
Hi, Jim.
Yeah, my name is Jim Hallbeisen, Judy, and my title is Director of Research. We're a small fertilizer company. We manufacture a hot mix material that farmers are using a lot of different ways. The main product, I would say, is what we call in-furrow, where they're putting the fertilizer right on the seed as they plant. Vegetable guys are using it in transplant solutions, tobacco farmers. And then we also foliar feed the product as the crop is growing. Our philosophy is that the plant goes through two stress points, or two main stress points, which are germination and reproductive set. And that's when we believe we want to get the fertility as close to the plant as possible. The other thing that's really coming in our direction is the pollution problems, Lake Erie, Gulf of Mexico, Chesapeake Bay. We have tried to get guys to cut their total input of fertilizer significantly. Like myself, I farm about 500 acres, and we're using probably less than half the nitrogen that a lot of our neighboring farmers are using, and we're very competitive with them yield-wise. And we think that's a biological factor. So that's the thing that we've really encouraged our guys. So we want to work with someone that knows a little bit about that and has some experience with it that can, educate us.
Well, I'm a test developer, so I learned what I could about soil because I have to know what I'm measuring and why it's important and things like that. But I'm not an agronomist. You guys are agronomists. And I think one of the things is, until now, to a large extent, it's been an art.
Mm-hmm. Yeah, no, you're absolutely correct.
It's not that I don't admire art, but if we can bring a little more measurement in,
Sure.
We'd like to do that. We know from the chloroform fumigation assay, 2 million articles, that this is the best indicator of soil fertility. And we do correlate 94% with carbon fumigation extraction.
Mm-hmm.
That said, most people are not aware that when your test goes to the laboratory, there's a 25% variation generally in the amount that the laboratory measures.
Mm-hmm.
So people are often worried about getting, variation in our test, but there is variation in any of these tests. Because, you're looking at a microenvironment. I mean, what I say is it's kind of like looking at New York City and looking at a bunch of apartments that are, maybe give you all the same square footage. But if you look at the number of people, in the same square footage and the same square footage, you're going to see something different. It's not going to be exactly the same. And that's because there's a lot of microenvironments and little things going on. So we do see more variation in pasture because the amount of organic matter can be quite different. And I think you're talking about biological activity, but we do not measure biological activity. respiration measures biological activity. And biological activity does not actually tell you, although Haney test and them indicate that it tells you the microbial biomass, it does not correlate with microbial biomass. In the rest of the world, it's only used for indicating stress. So what they do in the rest of the world is they measure the respiration by the microbial biomass. And if the respiration is high by the amount of biomass that's there, what you're actually seeing is organisms that are having to work too hard. So in cases like where there's toxins in the soil, you get a lot of high activity because the organism has to expend more energy to stay alive. Mm-hmm. If it's low pH, you get much higher respiration than you do from the biological activity. And the biological activity is actually turned on and off at different times in the soil. So, it's not as accurate as, we say field moist soil, because, the chloroform fumigation assay is done on field moist soil. Once you dry soil, we see losses between 20 and 80% of the microbial biomass. Mm-hmm. And when you try to reconstitute it by adding water, the way that the Haney test suggests you do, sometimes it will come back. Sometimes it will come back higher. Sometimes it will only come back to 20% of where it was. Mm-hmm. And what we've found is the fungal to bacterial ratio is not the same. That you're not regenerating the actual microbes that are in the soil. So you're looking at a different population. You're looking at a population that grows in your lab in a soil that's been dried. Mm-hmm. And more scientists are beginning to recognize this and to recognize the fact that PLFA has that real problem, that it works with dried soils. Okay. Mm-hmm. And the Haney test does. And so we've had some trouble getting as much validation as we'd like to do, because almost everybody I have a whole bunch of dried soils. I've had them for 10 years. Let's see if we can use them. Well, the answer is, you really can't. Okay. You have to do the test. The people who do the carbon fumigation, the chloroform fumigation or CFE assay, do it within a week of receiving it to get accurate results. Sure. And we need to test within 10 days of taking the soil. Some soils will stay the same in the refrigerator for a while, and some don't, but all the ones we've tested so far, we can hold them for about 10 days in the refrigerator. So it's not necessary to actually run the tests in the field. You can actually bring them in and you can run 20 tests an hour.
I see. Okay. What about the moisture? What about the moisture content of the soil? The spring of the year where the soil's still cool, your moisture is still, but then if we would go out there, say in August, do we have to concern ourselves with the moisture that's actually there when we get the soil to make your tests more accurate?
We do. And I'm not sure exactly why that is. I'm not sure whether it's because when they dry, they may become too stuck to clay and soil particles that they don't release, that people just don't see them. Mm. But chloroform fumigation, which costs $500, mm-hmm. has the same issue. So, people, that's been found over time. Mm-hmm. I think you're one of the guys on the forefront, because what we've said is, since chloroform fumigation was a $500 test, nobody was actually using it to predict what they should do or to evaluate.
For sure. That's right.
Just didn't happen. Now, I was inspired to start all of this by James Sotillo, and I think his stuff is still on the internet, and he was one of our founders. Unfortunately, he died at 52.
Wow. Mm-hmm.
He dropped dead of a heart arrhythmia while he was at a conference in Arizona. So, but he was, I was teaching and he came to the college where I was and he said, I need a test when I'm buying soil or when I'm evaluating whether the thing I'm working does. I need a test that tells me right away how things are going. I can't wait 2 weeks for a result. Mm-hmm. And he was sending things to Elaine Ingham's lab, and he was getting 50, 75% variation. He'd send duplicates. And because microscopy looks at much, much less than 1,000th of what we're looking at. Mm-hmm. Okay, so if you're looking at 1,000th less, statistically you're not getting a lot. I mean, they don't read very much of the soil. They take one little drop. They don't separate it from the soil. So they see very few microbes compared to what we see. We do correlate microscopy. So if you want, you can look at our samples on the microscope and you'll see the microbes. Mm-hmm. And strangely enough, they look pretty normal. Mm-hmm. I thought they would look much more ugly. I mean, we whisked them and everything else. The fungi look a little, kind of curl up on themselves and form ribbons and stuff like that. But the rest of them, are are very identifiable, and we don't have, soil in that. Mm-hmm. in that.
What our issue is that we know how our protocol works. Our idea with the farmer is that if you implement so much of our protocol, we can grow you a very competitive crop economically with significantly less inputs.
That's fantastic. That's the way to go. Yeah,
we're trying to figure out how to demonstrate that, and it's interesting that you brought up Elaine Ingham. In about 1996, maybe, when I first came with this company full time, what we did was we heard about her lab in Oregon, and my boss at that time said, we need to send some of our customers' soils out there. And then I want to go across the road to where the guys have been following the establishment, and then let her tell us what the difference is between them. And so we did that. We did several farms, maybe 6 or 8. I just saw the literature the other day, but we sent them to her, and she came back and told us, what she found. And then we firmly believe that the ability of the microbes to work in our environment is much better. That's why the guy can get by with significantly less inputs. And so we were thinking that she would say, well, your soil biology is much better than our competitors. And she reversed all of them. And so we just felt that she didn't have anything to offer. And so we have been kind of stymied since that time until you've come along now, and we're thinking that we need to hook our cart to your horse, and we're just looking for someone that can explain to us, what the measurement is showing us, so that we can tie that. We've got results. We're not concerned about that. We just need some help with explanation, because we think the answer is in the biological world.
Yeah, if the signal for the plant to send out exudates in the rhizosphere, which is 30% of its photosynthate,
sure,
is the need for nitrogen. That's the main one.
I gotcha.
It's probably also for phosphate and the others, but
Right.
So once you put that down, it basically doesn't feed the microbes the way it does, when you do it.
Sure.
So, what the trick is to not put down so much that you inhibit the plant from feeding the microbes, because the microbes in the rhizosphere and that area are basically the immune system of the plant.
Yeah. Like the human, correct?
Yeah, it stimulates the whole system. So without those, you don't really develop an immune system. So it's a human being who has no microbes, or an animal that's raised with no microbes does not develop an immune system, and neither does a plant. So the literature shows that, it's a huge study in Europe showing 97% less use of any type of pesticide in organic fields. Mm-hmm. That's not saying you have to go organic, but just saying that, we want to get away from pesticides. We have to stop killing off the microbial biomass. And I love the work of Clyde Kirkby in if you send me your email, Judy at MicroBIOMETER, if you email me, I'll send you his articles. Mm-hmm. He's just wonderful. And he's basically, in a lot of ways, doing what you're doing.
Mm-hmm. I see.
And he's gotten fantastic results. He's realizing that people just can't get away altogether from mineral fertilizers, but they can cut them back and they can mix them with other things, and they can come up with solutions that preserve. And the big advantage is, if you don't kill off the microbes, they're capable of storing some of that mineral fertilizer that you put down.
Absolutely.
So it won't wash away.
Yeah, no, that's right. That's stability. That's what we try to tell our clientele, that if they will work with it and bring it into their system, they're stuck to the soil. They're not going anywhere. And so, this significant loss that we're seeing in these water systems is strictly way too much, and it's toxic to begin with in many cases, and it's just gonna solubilize and wash away. We're putting this stuff on when there's no plant growing. See, the philosophy of fertilization has always been, well, you put it on, it rains, and it sucks it up. See, well, if you're putting it on and no plants out there, who's going to be sucking it up? It's going in the water system. See. So that's what we're attempting to show guys, and the cutting that level back that's a very big hurdle. I mean, the farmers have been brainwashed in, it might be the wrong word, but brainwashed into thinking that it takes pounds to grow bushels, and we've been able to demonstrate that is not necessarily true. We can do that significantly.
Yeah. We need a lot more people like you guys showing, working on things like that. And the other thing I find is, when people say to us, what am I going to do? We get asked that question a lot. So you told me, what this is, what am I supposed to do? And all I can say is, we can't tell you what to do, because it's going to depend on your crop and your cover crop and your soil and everything else.
Yep.
And, I've been advocating, and whenever I do these things, everyone says, where can I send it? But I've been advocating that it would be really nice if someone would actually do these experiments for the farmer.
Mm-hmm. Yeah.
You can see an increase in microbes within weeks.
Sure.
So, if you're putting down a cover crop that really is replacing something that's missing in your soil, you can see like a 600% increase people have within 3 weeks in a pot.
Yeah.
So, that doesn't mean that cover crop is going to go well with your crop.
Right.
Because the microbes it encourages may not be microbes that actually work well with your crop.
Mm-hmm. See, if you go back to the way my grandfather farmed, they had a few head of livestock and we had a basic rotation of forage, for the livestock. And then you interspersed a little bit of row crop in there. And, his utilization of inorganic fertilizer was minimal and still grew, very competitive yields. Now, yields have improved from breeding and that type of thing, but they were growing in some areas very significant yields with no fertility input or minimal fertility input. And see, the thing we try to stress to the farmer is, when you're harvesting a seed of corn, what is that? They want you to believe it's pounds of N, P, and K. Well, it's sugar. That's carbon, hydrogen, and oxygen. It's sunlight, air, and water. See, so the amount of fertility that's really necessary is not as large as they're suggesting. We got the tail wagging the dog, in my opinion. So this is something that we think that biological is really the correct answer. And we're hoping that with your test, we can go to, we've got some customers that, they're on the 4th generation using our protocol, and we'd like to go into their fields and pull that sample and use your test and then compare that to, the chemicalized approach and see what kind of difference the USDA numbers on what's adequate biomass and the ratios and that, we're not arguing with that, but we're going to establish our own because we feel that the guys having really good success economically and production wise, we want to see what those numbers tell us. And then we'll come up with some protocols to try to tell potential customers, when we stab your soil, you got this imbalanced or you're low. If you follow our protocol, you want to head in this direction. What we want to do is replace that chemical soil test. Judy, we want a test like yours that we can hang our hat on and have a lot of confidence in.
Yeah, well, I'd love to work with you to make that happen, so that's the kind of thing we need to do. I don't know if you have looked at on YouTube, on our website, we have some podcasts, some webinars I've done. Mm-hmm. And one is with Dr. White on rhizophagy.
Yeah, I haven't looked at that one.
Well, I think you'd really like it. He's followed up, and he shows his photographs are fantastic. Okay, and he shows some of them during the thing. What he has discovered is that when a plant is germinating, the tip of the root, the microbes actually enter through the tip of the root. Mm-hmm. And the plant gets 30% of its minerals from those microbes that it basically eats.
Sure. Okay.
Okay. And that without rhizophagy, without those going in, you don't form the root hairs.
Really?
Yes. And what happens is those microbes come in, they give up a lot of their nutrition to the plant, probably minerals and things like that. Mm-hmm. And then they are released through the root hairs. And he has pictures of them being released.
Really?
He has pictures of them going in. Okay. And this data is like spectacular. Okay. And so I think this makes a lot of sense because it's very hard to imagine how these microbes would have actually stimulated the immune system the way they would have without actually being inside the plant. And we do know that there's microbes all the way inside the plant. There's microbes everywhere inside the plant.
Right. Yeah. See, that's a concept that nobody really grasps,
Okay. And the other thing we know is there's 20,000 different antioxidants made in plants that are not over-treated with nitrogen and with mineral fertilizers. Okay. And these are the things, these are not counted when the USDA says organic isn't better. They're measuring sugar and carbohydrate and protein. Okay. Sure. And they say there's no difference. Right. Okay. These things have anti-cancer, anti-inflammatory, a lot of different things. And these are made by the plant that's stimulated actually by the microbes so that it protects itself. Right. Okay. So I think one of the concepts that a lot of people don't actually recognize is that a pathogen isn't necessarily a pathogen. Like right now, if I looked in your throat, I would see streptococcus. Mm-hmm. Okay. Mm-hmm. And your body actually uses those streptococcus to monitor streptococcus and make sure you don't get a streptococcal infection. Mm-hmm. Okay. Sure. And when we say you have a streptococcal infection, it's when the levels go way up. Mm-hmm. Okay. Yep. Now, maybe I don't know if this is of interest to you, but I'll just tell you what happens is these microbes communicate with one another. It's called quorum sensing. Quorum is if you have enough people to vote. Okay, so they send out a molecule. And when there's enough molecules there and they know the density is there, then they turn on the pathological proteins and enzymes that allow them to start causing damage. They don't cause damage up until that point. Mm-hmm. And so your normal microbes are keeping those other microbes in check. Correct. Once you get rid of those normal microbes and you don't have them there, you can't keep these guys in check. Sure. Okay. And the normal guys have so many ways of keeping them in check. They make like antibody-like things that bind the quorum sensing molecules that they send out. Okay. They have all kinds of little tricks. They have enzymes that can break down the pathogenic molecules that are being put out so that it can penetrate the cell and do things like that. So, you really want, first you want microbes there and being eaten so that you develop good root hairs, which are associated with, you'll see on his pictures there, with much more rugged root development than you'll see without root hairs and without the microbes there.
Sure. That's another aspect of us using calcium in soil is to soften that soil so that the plant can proliferate that root zone. Our analogy to the farmer has been it's the foundation. It's really the key to the plant's success. And some of the so-called experts are saying, well, they only absorb a little bit of nutrition and it's all in the topsoil and that. And really, we're advising our people, we've followed our protocol on guys' farms for 5 or 6 years, and we get a backhoe in there next to a corn plant and we'll find roots, 60 inches in the ground and see. Wow. That's we feel that's very critical. Number one, it gives it anchorage, it gives it access to water, it gives it access to mineral, but you're also, which we didn't realize, is you're feeding biological life to that depth too. See, so
yeah, and you're storing carbon.
Yeah, absolutely.
And you're preventing compaction.
Yep.
And stuff that's what we need, is that, and the more root you have, the more mycorrhizal fungi you can have, and a mycorrhizal fungi increases the root surface area up to 1,000-fold.
Sure, exactly.
Now, it can help a seed if there's mycorrhizal fungi on it, if it's the right one, the mycorrhizal, and the right mycorrhizal for the soil that you're putting it in. Okay. but I think more and more people are saying your soil has its own mycorrhizal population,
Mm-hmm.
and that endogenous population is the one that will do the best for your crops.
Yeah. We deal with, for a lack of a better term, what we call bug in a jug companies that are selling, microbes that the farmer, puts in the soil. We're not, it's very simple. What's it cost you? What kind of payback do you get? Our issue is we have to create an environment that is conducive to the proper microbes to build the symbiosis with the plant. And that's more critical than purchasing that year after year after year. Build your own population within your soil by using the correct protocols. And
I think that's exactly what all the experts are saying finally. And I've looked at how many microbes they have in that and how you dilute it and you put it down. Okay. Yeah. And it is, the analogy I have is, it's like dropping 2 Americans into China and say they're going to multiply and take over.
That's excellent.
I love it.
Now, one of the things that I discovered going to some of these conferences is that these guys, if they're selling a biological, they don't have to say what else is in the solution.
Absolutely.
So probably the biological is just an excuse for whatever they're doing. And a lot of them, it's just plain sugar.
Yeah, well, that's right. That's exactly right.
And you can get a boost with sugar, but that's actually not good for your soil.
Very excellent. We are in total agreement.
It basically is like giving a kid a candy bar right before a 3-year-old boy a candy bar right before bed.
You're gonna regret
it. We've always told our clientele base that when you went to grandmother's on Sunday, before you got the apple pie, you had to eat the lima beans. And that we think it's very important to feed the biological correctly and create that proper environment, in our opinion.
That's what we say is feed and create an environment. That's what you need to do. Yep. I mean, okay. And these microbes have all these antibiotics and all these tricks, and they're defending their territory.
Sure, absolutely.
That's how they keep this balance. So the idea that you can put these down and, they're going to do. So we thought when we first started that the amendment companies would buy us. But that amendment is probably only going to work at best 10% of the time.
Exactly.
So that means that if this guy recommended our test, 90% of the time, 90% of his clients would be very disappointed.
Yeah, no, that's right. That's exactly correct. See, our deal there is they how much repetition do they want? I mean, the fertilizer companies, they want to sell more next year. So with the bug in the jug, they want to sell more next year. And our goal is to wean our clientele off of that the product you're raising is mostly sunlight, air, and water. So we don't need a lot of additional amendments to be successful. You just got to work with Mother Nature and the way she's designed this is perfect. Now let's get it to work the way it should work.
Right. Absolutely. I think most people now feel that most people are going to probably need to use supplemental nitrogen, phosphate, and potassium and other things because we're not, in nature, that what we just grew would never leave that field.
Right. No, that's right.
And we just keep taking it away.
Yeah, that's right. There is removal and we will need to remove some of that. But again, the biggest volume is sugar that you're removing. So what you actually need to replace what's removed is much smaller than what they're suggesting. And see, the the pollution is telling us that we're adding too much back, and we have to get that pared down and get it utilized rather than just throwing more.
And I think one of the things that, that science has told us is that one of the big builders of carbon and nitrogen and the things that you need to encourage microbes is the root mass.
Yep, exactly.
Okay, and that's the biggest contributor, and it's a big contributor to carbon sequestration.
Oh, yeah. Well, see,
you're building good root mass.
Yep.
Root mass tells you that you're doing a really good job at fertilizing because those roots are not removed.
That's correct. Well, what we've been able to demonstrate with our program that when they start following our protocol, that the health of the plant, their animals respond to that. We see much better growth, breeding, and all the health benefits. Vet calls go down significantly. And, we're saying to the farmer that the health of that plant is improving because you're working within the system. And the deeper that you get that root mass to go, you're insulating yourself against stress points, lack of rain or too much rain, if you can get the water out of there so that the roots can breathe again. This is a very big factor.
But once you have the roots there, you have the arbuscular mycorrhizal fungi, and they make glomalin, and the glomalin is one of the big contributors to soil carbon, and it's very sticky and everything, and it's what gives soil its structure.
Stabilizes it. So, you can hold 50, if your microbial biomass is healthy, your soil can hold on to 50% more water. So you're 50% less, against, having problems with drought. Exactly. No, that's right.
And the arbuscular mycorrhizal fungi, actually, they stimulate the plant. And so what you see is you have a root that's like this size, okay? And during a drought, the arbuscular mycorrhizal fungi colonized root, it swells up like this with sugar. It's stimulated by the fungus. See, osmotic pressure goes up, and the root pulls more water in against the gradient. So the plant is much more able to survive drought and things like that.
And we see that in just the appearance of the plant. See, that's one thing that we've been big on with potential clients is when we look at your crop, everybody, we call it windshield farming in our operation. You want to go up and down the road 80 miles an hour and look out the windshield and see how good the crop is. We're saying you got to get the shovel and start digging around that root zone, and that will tell you more about the health and vitality of the plant than any visual observation that you may have. So
I think you're totally right. And I've seen like pictures of fields when you have a good root system and a wind comes up, the plant survives. The other field is all laying all flat and everything. So
with that derecho that they had in Iowa last summer, we had a little bit of experience with that. Some of our good customers were in that band, and they did have damage, but it was not to the same extent as right across the fence row. And we attribute that to root zone.
That's. Did you publish that information, or did they do anything?
We try to do some of that. It's again, with our protocol, we've been pretty well castigated by the establishment because, we're advising to do opposite of what the establishment has been doing for a long time. And
we had the same problem.
Yep. We appreciate your position, believe me.
I mean, Haney's group runs the USDA.
Yep.
So when we first developed this test, it was done on a USDA grant, on an NIH grant. I had $600,000. It was to set up quality control for pharmaceutical companies, actually, to train people to do that. And then I met James Sotillo, and he said, this is a test we need. And I'd been developing tests before that. That was my job. I did medical diagnostic testing. And so I started work on it. And Haney's group was put on the panel. And the guy just said, was Franz Lubers. He said, I tried that. It can't be done. So then I sat with him at a table at a conference at the Soil Institute. And I said, try my test. He said, I wouldn't bother to try it.
I believe it. Yeah. We understand.
Wow.
It's just like, but we're working more and more with USDA people. Mm-hmm. And right now we're working with one of the leaders in regenerative, which is Janice Tice at Cornell. Mm-hmm. She's doing a big study with us. And we worked with Dr. Ketterings at Cornell. We just finished some studies with her. Mm-hmm. And we're working with University of Tennessee with several departments there, and they're USDA people. So there are, Janice Tice is like, she's like the lab at Cornell won't collaborate with her.
Mm-hmm. I believe that.
Because, she says you need, and she's saying like, what I'm going to do with the study she's doing, okay, is she's comparing our results on 125 plots with the Cornell soil health test. Mm-hmm. And she wants to see which things we correlate with.
Mm-hmm. Sure.
And she's pretty sure that it's going to be soluble organic carbon.
I see.
We've been able to show over time, like if you look at a field that's been treated the same way for 30 years compared to another field, you can see that, regenerative type practice led to higher microbial biomass, and it correlates very nicely with soil organic carbon.
I see.
But let's just say that you just put down a good cover crop, and it increased your microbial biomass by 300%. So you went from 100 to 300.
Okay.
You didn't increase your soil organic carbon by 300%. Mm-hmm. It takes 10 years to increase your soil organic carbon to where one can measure it.
I see.
You can only, when you're doing that, the only way people know if they're actually increasing their soil organic carbon is if they've increased or maintained the microbial biomass that's been sustaining their good practice.
Right. Exactly.
Okay. Because 60 to 80% of soil organic carbon is called necromass. That is the bodies or the remains of dead microbes.
Right. Yeah. See, that's something we've been very concerned about is, that the chemical testing labs are saying, well, you just measure organic matter and that's going to tell you what you're doing with your microbes. And we're not buying any of that because we've got way too much field experience to show us that that's not true. And so that we're not buying what they're saying. And see, this is why we've gravitated.
I hear it more and more. And there's a lot of people talking it now. And I was working with a young girl in Texas, and she was like, she said, they told me I have no magnesium, I have no potassium. She said, but my plants have plenty of these things. How can this be?
Yeah, that's right. Exactly.
And because the microbes can make those things available.
Yep.
They mine them, and they're just not available for testing.
Yep. That's right.
For measuring.
Exactly.
Chemically.
Yeah.
No,
Yeah.
you're definitely on the track of where we're going. And most of our sales are out of the United States.
Yeah.
Like Australia, which is way, way ahead. We do a lot of sales there.
Sure. Well, again, I think the entrenchment of certain companies it's going to make it more difficult, and that's our experience. But that's of no concern to us because our only allegiance is to our clientele. We're trying to make them all as rich as they can be. And then we want to give them good information and see this is something that we're really looking for is some help with this testing of soil to show this differentiation, because there is a lot of, farmers are figuring out that just pouring on fertilizer is not paying the bills. And see, this is plus with the pushback on the environment, that creates another regulatory thing that can be problematic for them too. And we're getting feedback through our customer base that even some of the major corporations are looking for particular tests to show that they're buying produce from farmers that are using a specific protocol. And Cornell's, like the tests you mentioned before, these companies are looking at these tests if they've got possibilities. And if yours does what we think it'll do, you're on the front end of something big, in our opinion.
Well, University of Tennessee is having it published, but they have these lots that have been treated for 39 years. So we tested 144 lots like this. And our test was the only test that actually measured soil health.
Okay, great.
The soil health test from Alabama, Indiana, the Cornell soil health test, PLFA, it's all written down. None of them was able to actually predict soil health.
That's big.
Okay.
Yeah.
And then, what they tell you is the soil health test is very dependent on the type of soil you have in an area. So the Cornell soil health test is ideal for northeastern United States. It does not work in Tennessee or Alabama or anything. So under their guidance and everything, we worked with them on the validation against CFE, chloroform fumigation, and we had samples from all over the United States. So that's why we did that, so that we could qualify that.
I'd love to interject in here. Sorry, Jim. I hate to interrupt you guys because you're having such a great conversation. But I know, my background, you can probably guess since I'm more quiet, is not as strong in Jim's as farming. So, one question I had that came from our farmers that I want to make sure we take the time to ask is really, how can our farmers take better soil samples? How can we make sure that they're getting a really good sample? What's the best method for that sort of thing?
We ask people to just take a soil from the top, 3, 4, 5 inches, just the top. Okay. The microbes actually, that is the microbial population from which the rhizosphere comes and everything. And that's where you see them. Okay. And what we believe happens, and it makes sense actually, is that that the microbes that are most susceptible to dying after you take a sample are those that are rapidly dividing. So those are the ones that are basically biologically active, because the microbe, when it's active, it's just dividing. It doesn't get larger.
Right. Mm-hmm.
It just divides. So dividing cells are very sensitive to stress.
I see. Okay.
That's how we kill cancer cells. We use toxins that kill rapidly dividing cells.
Oh, sure.
So it's not a surprise. So basically what happens when you've collected a sample and you haven't put it in, we say put it in a plastic bag. Okay, don't lay it around in the sun. Don't mistreat these microbes. Think about, how you would like to be sent to the lab.
Sure. So most of the people that are working with your test, are they're taking the stick or a probe, whatever they refer to it, and then you put it in a plastic bag and you'll do your probes and then you'll go to a spot and run the test is what.
Yeah, you can run it on a desk or something. If you want to do a bunch of them. When I do, we're working with one client now, Riomax. I don't know if you know Riomax.
No.
They work just with cows.
Mm-hmm.
Okay, so my husband has ponies. So for his birthday, I bought him some of their Riomax stuff, which is supposed to be very good. Mm-hmm. So he went out and he collected 36 samples from the field before he goes out and sprays with it. So we're going to do a little experiment and see, what does it look like before you put the Riomax on and after you put the Riomax on. And then wait until after it's rained so it's in for the ponies. So he just came in and he had, we have a sifter, a big sifter, so we don't use the tiny little one. You can buy them on Amazon, like $10. And sure. I put it in a cake pan and then it goes really fast.
Right. I
just sift them all up a little bit because you just need a half cc. And, I set up the tubes and you run, once you get used to it, you can run 10 at a time. I run 10 at a time. And I highly recommend that you use a filter. I like the filter. You can use a filter and cut the time to 10 minutes.
Okay.
We're probably going to implement that. It's more expensive for us. These filters are very expensive for us. They're like 30 cents a piece. So
what kind of filters are you using?
Plastic. But if you have extra time, the filter just gets put into the tube. It's a serum filter.
Sure. Okay.
And so the fluid that comes up is here, and the microbes are going to precipitate over time. So timing is kind of critical. Okay. But once you put the tube in like that, you can test it for up to an hour.
I see. Okay.
You shake it a little so you get them back into solution because all you'll have there is microbes. So, it's nice for doing a bunch of them. So you're not like, oh my God, I have only a minute. The phone rang.
Sure. No, that's right.
Yeah. And then you're not out in the middle of a field, doing everything. And your lighting is, the same lighting every time and everything. It's more convenient to do that way. Sure. So I keep telling people, don't call it in fields, call it on site.
Yeah, I see. I understand.
So, because it's much more convenient to do it, there. And, if people are mailing at this time of year, we're collecting samples right now from people. So we're doing, every once in a while, we try to reevaluate everything and tell people, are they getting the same numbers we're getting? So we're telling people this time of year, if you priority mail a sample the same day you take it, you could mail it to us.
I got you. Okay.
In the summer, but I don't know, I've heard there's a lot of problems with the post office right now.
Oh, it's been a disaster. So. Really? Oh, yeah.
Oh, yeah.
Yeah. We've struggled in our business with the post office tremendously.
So what are you guys doing? Are you going to UPS?
Well, we are, it's more with receiving payments and that type of thing. That's just been, and yeah, some of our guys have actually done that, gone to UPS sending us that stuff. It's just, sometimes it's been, we thought it would relieve significantly, and it is better than what it was back in December, but November and December were just a disaster. So.
Yeah.
It's very slow. Very slow.
Yeah.
It's longer than usual.
No, I've got another.
So you just have people to use a Ziploc bag for collection.
Mm-hmm.
You know.
If we were to interact with you personally, I looked at on your website. Do you live in Orange County, New York? Is that correct?
Yes. I have a home in New Jersey too, but now I'm trying to live here just in Orange County only.
We have
Because
a significant amount of business.
I just can't manage to.
In Orange County, and I would be in that area. I'd certainly like to.
I'd love to see you. Yeah, you could Google us. We're on a farm.
Okay.
We'd love to see the ponies.
Oh, my husband has 23 ponies.
We'd love. Yes. Please.
No, I was in that in my youth. I will turn that over to him. That's it.
And his main interest is in training them.
I see.
Oh, yeah.
Having failed to train a wife and 3 daughters, I can tell you he's having no more success with ponies.
Oh.
I understand.
He's trying.
He's too sweet.
Absolutely.
I love it. Well, we would love that and love to visit and keep up that relationship.
I would love to have you come and see, things, because the lab is in our basement. It's a big basement, It's 9 feet tall and everything. And then we have more space in the barn. There was a apartment. We do we do assembly and stuff like that of the kits over there. So we're in about 4,000 or 5,000 square feet here.
Sure. Great.
And,
That'd be great.
We'll probably try to be moving out because we have trouble getting internet up here.
I see. Okay.
Right now we use hotspots. I
got you. Okay.
And we're right by an AT&T tower, so the hotspots work pretty well. But a big part of our, the phone thing is run by Dr. Brady Trexler, who's, developed how to read the card and everything, which is, absolutely key to,
sure.
our success and everything. And most of the time he doesn't get enough power because he's working all the time on the internet. So he has to work, at a distance and everything.
I got you. Okay. That's on our agenda. We're tinkering with it. We're at a time frame now that, like myself, I'm going to be going to the field here pretty soon. And but once the planning and everything is under control, and see what we have are individuals that rep for us around the country. Our gentleman in New York lives up in the Finger Lakes area, but he has a significant amount of clientele in Orange County. And so I would interact with him. And then if he had clients that he wanted to interact with, we would do that. But if we can get close to you, we certainly would like to sit down and chat with you if that's possible.
That'd be fabulous. I'd love it. Now, one thing is, if you have samples from all over the United States, that's what we're looking for now for this, we want to check out all the different soils and see how they
Sure.
run. And everything.
Sure.
So, and we'll be doing microscopy on them and everything just to make sure we're still,
Okay.
So if you had samples, you could ask people to send them to us and we would test them and give you the results and you could give them back.
Okay. Well, that's something I would like to discuss with you. Right now we're getting our feet wet, with the test itself. We've got some ideas on things we want to try to do, and it's just not with soil. The compost tea thing. We have a lot of clients that are working with compost teas. And the other thing that we're looking at is these bug in a jug deals. They're just, their marketing is just very, very intense, and farmers are believing that they're on the right track with it. And we're not so sure about that. And we're thinking this might be another way to kind of, put a little wet blanket on some of that stuff, is to see, is there anything in there that they say is positive? So,
well, I'll tell you, the story that will tell you you're right. Okay. Indigo Ag.
Yeah, I've seen their stuff. Yeah.
Okay. Well, Indigo Ag they've raised probably $200 million based on coating seeds and providing microbes.
Right.
Okay. That's not what they're talking about anymore.
Really?
Mm-hmm. It worked when they were in Tennessee on some fields. Okay. Now what they have is selling people fertilizers and all kinds of stuff.
Mm-hmm.
You buy all your supplies from us, and we'll guarantee to buy your crop. That's their business model now.
Really? That's pretty interesting.
So they're holding the crop so that the farmer who needs the money at the end of the season knows that he gets a fee, and they're taking the interest and betting that they can get more money for it in March.
Yeah. No, that's right. Well, the co-ops have been doing.
They switched from this whole thing. You don't hear anything about the bugs anymore.
No, that's right.
Because, they'll work some of the time and they won't work all the time.
Right. And we've been privy to that. I worked with a biological bug in the jug back in the 80s. And just what you said, the idea was that if you put this in furrow with your fertilizer, you can cut your nitrogen usage back significantly. And we did test plots with it. It worked very well that first season. We came back to next season and it fell flat on its face. And another season, it was the same thing. So we just were convinced that it's a nice place for a con job, in our opinion. So, and
if you're a microbiologist, you start out and you put a bunch of microbes together.
Mm-hmm.
You put them all together, then you grow them up. Okay. You won't get the same ratio of everything every time.
Mm-hmm.
One will usually end up dominating everything.
Right.
So, they come up. So biomakers, we're talking of working with them. They do much more complicated, they're trying to look at, they're kind of in research. They're looking at what combinations and everything of microbes would work on a granular level, like which things. And we were talking to him just last week, and he said, they don't have the microbes they say they have. We've tested.
Wow.
That's not what they're selling. They say it's certain microbes. And they said like they could say it's 10%. It might be 90%.
Right. That's right. They're not monitored at all. So
no, they have no monitoring at all. It's snake oil.
Yeah. No, that's right. Absolutely.
So I don't, there's a whole bunch of people that go to the health store every week. I play tennis with some of them. Every week it's a different treatment that's going to save you,
Absolutely. I understand.
Well, I don't want to take too much of your time, Judy, but we so appreciate this. I know we could talk to you forever, especially Jim. He's like, oh, somebody who sees things the way I do, right? I'm sure he's so thrilled.
Well, I feel the same way.
Oh, good. And this is something we're all really passionate about. So we're just thrilled to have found you and to have met you and gotten to interact with you and learn from you. And we really appreciate your time and definitely want to come visit and spend more time, interacting with you and picking your brain and sharing our experience and our thoughts too. So I think that'll be wonderful. But I do not want to take too much of your time. And I know we're a little over our allotted time frame already. So I do want to cut things, but
okay.
Thank you so much for your time. And I want to give Jim, and you a moment. Do you have any final questions, points you want to go over really quickly to wrap up?
Well, you have my emails. So, Jim is free to call me anytime. Well, like our ideas for like where we go next and what we do and what we need to do to get better, we need input from people who are, passionate about the same types of things we are.
Correct.
And in our field. So, I think for any of us who are in this field, education and educating our users is our biggest goal.
Yep, that's right.
Absolutely.
Exactly.
So we always feel like people are very free to use any of the material I have up on ResearchGate or any of the materials we have. Mm-hmm. And so, and modify them and just, let's get the word out there because,
sure.
And Laura's been doing a lot of work on this stuff. And, when I do the webinars or podcasts, what she's found is the more technical we get, the happier people are. And, that's what people didn't think.
Right.
Was going to go down. But there's people out there who are saying, I started with saying, like, what does it mean? Like, let's really explain to people. I have a video up there I think your people might like about actually how the immune system of the plant, the microbes actually protect the plant. So I go into actual specific instances of how it works.
Sure.
Because just to say something's healthy doesn't really make the connection.
Right. No, that's correct. Do you have a video that explains the basics of your test itself, how it works?
I don't think I have, actually. I should get that. That's a good idea.
That's something our guys would be very interested in. That we're introducing it to them. And particularly guys that have been with us for a long time. And, we're going to say, we think we've found something that is going to give us this kind of information, and they're going to be interested in how that works. And your pedigree is a big part of that too, ma'am. In other words, they've been around the horn with people that don't have the kind of experience that you've got in developing tests and that type of thing. And I think that background is really important for our guys to understand that, we think we've got something here that's going to be very helpful to us.
Yeah, I think one of the reasons we could do it was, medical diagnostics is way ahead of soil testing.
Oh, yeah.
There hasn't been a new soil test for 100 years, practically.
Oh my gosh.
That's right.
So, one of the things is, bringing medical diagnostic technology, because I have 12 patents in medical diagnostics.
No, that's right. Exactly. That's why we're talking.
Okay, thanks.
That's very impressive.
Yeah. Actually, worldwide, they just told me from Syracuse University the other day, I have 165 patents listed.
Wow.
I don't know, they were, they patented them all over the world. So they count, to me, I just say US patents, but they patent them in a lot of different countries and stuff.
Yeah.
So, that's just what I've worked in, all my life. So, or most of my life. So it's a passion of mine.
No, we want to get educated. That's what we're after.
Okay. So I really recommend that you look at on our YouTube and look at the one about it. I don't actually talk much about microbiometer at all in it. So I try to make these just scientific.
Sure.
So the two on rhizophagy and the one on amino acid on the immune system, or how plant microbes protect, I think your customers would really find them useful.
Okay, very good. We'll get to the bottom of that.
Made a note on it, so we will look them up. Thank you.
Okay.
Thank you very much, ma'am.
Thank you, guys. It's been a pleasure meeting with somebody who's in, on the same page.
Exactly. I understand.
Thanks for listening. If you enjoyed what you heard today, please subscribe and share with a friend. If you'd like to learn more about the Growers Program, visit our website, www.growersmineral.com.